Cellular respiration is a process that converts oxygen and glucose into carbon dioxide, water, and energy in the form of ATP. Aerobic cellular respiration has 4 steps: glycolysis, pyruvate oxidation, the citric acid cycle, and the electron transport chain. Through the 10 enzyme catalyzed reactions in glycolysis, a glucose molecule is split into 2 molecules of pyruvate. In the first step of glycolysis, a glucose molecule enters a cytosol and receives a phosphate group from ATP in a phosphorylation reaction. It now is glucose 6-phosphate. Then it undergoes an isomerization reaction where it becomes fructose 6-phosphate. And then another phosphate is added from ATP and another phosphorylation reaction in step 3. The molecule is now fructose 1,6,5-phosphate. In step 4, there's a lysis reaction where this molecule splits into G3P and DHAP. In step 5, there's an isomerization reaction where DHAP becomes G3P. Now 2 molecules of G3P enter step 6. Step 6 is a redox reaction where the G3P molecule gets oxidized as it loses the 2 electrons and 1 proton. NAD+ accepts this electrons and proton, and the second proton is released into a cytosol. Additionally, a phosphate from the cytosol is added to the molecule. The resulting molecule is 1,3-biphosphoglycerate. Step 7 is a substrate-level phosphorylation reaction where one of the phosphates is transferred to ADP to make ATP. We now have 3-phosphoglycerate. 3-phosphoglycerate enters step 8 where there's a mutase reaction. This is when chemical group moves somewhere else onto the same molecule. The phosphate group is moved from carbon 3 to carbon 2, and we are given 2-phosphoglycerate. And then step 9, electrons are moved from one part of the molecule to another, and a molecule of water is also removed. This leaves us with phosphoenol pyruvate, otherwise known as PEP. Then in step 10, a phosphate group is removed and given to ADP to make ATP and substrate-level phosphorylation reaction. This leaves us with a molecule of pyruvate. Now, we must remember that starting step 6, 2 molecules of G3P entered step 6. That means this process occurred twice, and we are actually left with 2 molecules of pyruvate. While glycolysis occurred in the cytosol, the next 3 steps are going to occur inside the mitochondria. In order for pyruvate oxidation to occur, the pieces of pyruvate have to pass through the outer and inner membranes of the mitochondria. They pass through the outer membrane through the large pores, and then they pass through the inner membrane through the help of specific membrane carrier proteins. The first step is a decarboxylation reaction where a carboxyl group is removed from the pyruvate and forms CO2. This is approximately where 33% of our CO2 comes from. Step 2 is an oxidation reaction that involves the oxidation of the 2 remaining carbons and the production of an acetyl group. Dehydrogenation removes 2 electrons and 2 protons from the acetyl and transfers 2 electrons and 1 proton to NAD+, making NADH, and the remaining proton gets released into the ion solution. In the final step, acetyl reacts with sulfur on coenzyme A, and this forms acetyl-CoA. This molecule is now ready to enter the citric acid cycle. The citric acid cycle consists of 8 enzyme catalyzed reactions, 7 in the mitochondrial matrix and 1 binding to the matrix side of the inner membrane. In the first step, coenzyme A detaches and the acetyl group joins oxaloacetate. In the second step, there's an isomerization reaction where citrate, the product from the last step, forms isocitrate. Then isocitrate is oxidized, a CO2 is removed, and NAD+ is reduced to NADH. In step 5, alpha-ketoglutarate is oxidized by NAD+ to become succinyl-CoA and NADH. A CO2 is released and a coenzyme A is added. This forms succinyl-CoA. Succinyl-CoA releases the coenzyme A, and the energy from this turns GDP into GTP, and this in turn turns ADP to ATP and substrate-level phosphorylation. Step 5 produced a molecule called succinate. In step 6, succinate is oxidized as 2 electrons and 2 hydrogens are removed and added to FAD, which forms FADH2. This forms fumarate. A water molecule is added to fumarate to form malate, which enters step 8. Malate is then oxidized to oxaloacetate as 2 electrons and 2 hydrogens are removed, and oxaloacetate can now react with a new acetyl-CoA and start the cycle all over again. In the electron transport chain, NADH donates a proton and an electron to complex 1 and becomes NADP+. The electron makes complex 1 supercharged and has energy to pump protons into the intermembrane space. It also pumps other protons from the matrix to the intermembrane space. The proton gradient now begins to form. The electron is now given to UQ from complex 1. After that, FADH goes to complex 2 and gives it its electrons and becomes FAD. Complex 2 can't pump the electrons, so it gives its electron to UQ. UQ now gives both of these electrons to complex 3, who gets supercharged and pumps hydrogens into the intermembrane space. This also contributes to the proton gradient. Complex 3 then gives the electrons to a complex called cytochrome C. Cytochrome C passes the electrons along to complex 4. This supercharges it, and complex 4 pumps hydrogens into the intermembrane space. Finally, complex 4 gives the electrons to oxygen. The oxygen is now split into 2 ions, and the protons are added to make 2 water molecules. The ATP synthase molecule uses the proton gradient, and through the process of chemiosmosis, it forms ATP. 3 phosphates bind to the headpiece, and it rotates in a way that catalyzes the formation of ATP from ADP and an inorganic phosphate.